Hello, my name is Chris Ray, and I'll be talking about the mutualism between whitebark pine and Clark's Nutcracker. This mutualism is famous for structuring high-elevation ecosystems throughout western North America, but I'll focus on forests of the Sierra-Cascades region from northern Washington state to southern California. This research was conducted through a collaboration between the Institute for Bird Populations and the National Park Service, and my collaborators are listed here as co-authors. Questions or comments can be directed to me at the Institute for Bird Populations. A beautiful illustration by Misaki Wichita depicts the many roles of whitebark pine in high elevation ecosystems and really emphasizes the importance of Clark's Nutcracker in facilitating the impact of these trees. Notice the different species foraging on whitebark, and especially the interaction of snow and whitebark on the slopes in the background. As a keystone species, the presence of whitebark increases biodiversity, structures ecosystems, and alters hydrologic cycling. So this tree literally provides water, forage, and habitat for many other species. And Clark's Nutcracker controls the distribution of these processes by dispersing whitebark seeds and helping them germinate. Nutcrackers store more seeds than they eat, and they cache them in microhabitats that help the seeds grow. Whitebark feeds this mutualism by offering up a very nutritious seed, and most whitebark germinate from the neglected food stores of nutcrackers. But there are new threats to this mutualism. Whitebark is being killed by an exotic rust fungus and by native pests and wildfire as the climate changes. This image shows the lethal effects of white pine blister rust as it girdles a whitebark pine. Our study focuses on effects of blister rust in Washington state, where rust is now common, and in California, where it's not yet so prevalent. As whitebark are killed by blister rust and other threats, it seems likely that nutcrackers will also begin to disappear from these landscapes. If so, there would be fewer nutcrackers to disperse whitebark seeds, leading to a vicious cycle of increasing whitebark losses. But Clark's Nutcracker feeds opportunistically on many resources. In this image, a nutcracker forages in the crown of a fire-killed ponderosa pine. So our main question in this research is whether nutcrackers actually respond numerically to trends in whitebark as a resource. We studied this question in 4 national parks. In Washington state, whitebark and nutcrackers have been monitored in North Cascades and Mount Rainier since 2005. In California, Yosemite and Sequoia Kings Canyon have been monitored since 2011. Our tree and bird data come from separate monitoring projects with sampling plots that don't typically overlap. Bird counts are conducted at points along transects, which look like squiggly lines on this map of Sequoia Kings Canyon. Tree data are collected from plots shown as triangles. So one goal of our project was to develop a modeling framework that can integrate data from different monitoring projects and across these 2 trophic levels, from seeds to seed predators. We also explored separate spatial and temporal models. For the longer data sets from our northern parks, where whitebark are clearly in decline, we aggregated data at the park scale and modeled annual variation in both trees and birds. For the shorter data sets from our southern parks, where whitebark are not yet in decline, we aggregated data over time and modeled the spatial distribution of nutcrackers as a function of tree resources. An interesting feature of our southernmost park is that whitebark gives way there to another 5-needle white pine. As we move from north to south within Sequoia Kings Canyon, foxtail pine becomes more abundant than whitebark. Foxtail pine has a much smaller seed with its own wing for dispersal, although it's also harvested by Clark's Nutcracker. So we modeled Nutcracker density at each point in this park as a response to the local abundance of whitebark and foxtail. Tree cover within 125 meters of each bird point count station was our metric of local tree abundance, and we allowed for an interaction between effects of whitebark and foxtail on Nutcracker density. We used Bayesian hierarchical models for our analysis to allow for imperfect detection of birds and effects of covariates like background noise, date, and observer on the distance and time to detection of nutcrackers. These models also allowed for any uncertainty in tree data to enhance the uncertainty in estimates of bird density that depended on tree metrics. Our main question was whether nutcrackers respond to trends in whitebark, so I'll focus here on the submodels of nutcracker density that we considered. In the temporal models that we applied to data from 2 northern parks, our null model was that nutcracker density just varied by year. We also considered several other models in which nutcracker density responded to annual variation in whitebark metrics like the number of live trees, rust-infected trees, or total crown kill of whitebark at the park level. Crown kill was defined as the mean percent mortality of whitebark crowns. Our expectation was that nutcrackers would be more abundant where more trees were alive with healthy seed-producing crowns. In the spatial models that we applied to data from 2 southern parks, our null model was that nutcracker density just rose with elevation. In Yosemite, we also considered a model in which nutcracker density responded to whitebark cover. And in Sequoia Kings Canyon, we considered effects of whitebark cover, foxtail cover, and their interaction. Starting with spatial results from Yosemite, this plot shows that nutcracker density was higher where whitebark cover was more abundant. Whitebark cover and elevation were highly correlated, though, in this park, so 2 models were supported by the data, effects of elevation or effects of whitebark cover on nutcracker density. In Sequoia Kings Canyon, whitebark and foxtail cover were less correlated with elevation, and we found support for additive effects of all 3 variables on nutcracker density. There was no support for an interaction effect between whitebark and foxtail. So nutcracker density was at least somewhat predicted by whitebark cover in all our spatial models, although foxtail cover and especially elevation explained additional variance in nutcracker density. The results of our temporal models of nutcracker density varied between the 2 northern parks. In North Cascades, nutcracker density varied dramatically by year and was not explained by whitebark metrics. In Mount Rainier, nutcracker density as a function of year is shown in gray, and nutcracker density as a function of crown kill is shown in orange. It's clear that nutcracker density declined dramatically in this park, but because crown kill trended with year in our model, it's not yet clear whether year or crown kill was the better predictor of nutcracker density. In summary, we found that white pine blister rust has had its largest effects on whitebark pine in Mount Rainier, where nutcracker densities have clearly declined. Nutcrackers that used to forage in Mount Rainier might have left this park for outside resources. At the same time, nutcracker densities have fluctuated more widely than whitebark in North Cascades, in a pattern that suggests these birds are tracking resources other than the park-level population of whitebark pine. These birds can move long distances to forage on a variety of resources, perhaps leaving the remaining whitebark without an effective dispersal mechanism. As these monitoring programs continue, we hope the flexible modeling framework we've demonstrated will be used with additional data to determine how nutcracker densities continue to respond in time and space to whitebark and alternative resources. Thanks for your interest in this project. I'd like to acknowledge again my co-authors at the National Park Service and the Northern Rockies Conservation Cooperative, and welcome questions or comments through the Institute for Bird Populations. My email address is cray, C-R-A-Y, @birdpop.org.